I assure you that what you will observe is a vast wasteland.
Free and uncorrupted communication.
I'm Chuck Samuel, and I'm here at UCSB. I'm a biochemist and molecular biologist, and I'll be moderating the first technical session that deals with novel systems for the discovery of new therapeutic targets. We've seen remarkable progress over the last few years in developments of new technologies and new strategies to address important biologic problems. And the very beautiful talk that we just heard illustrates that point, beginning with basic phage virology and the development of phage display and the utilization of that technology to gain really exquisite understanding of specificity of interactions in biologic systems. There are a number of examples where we've had gains in technologies that allow us to get greater insights into important biologic problems, and we'll have 5 speakers in this session addressing these sorts of points, ranging from novel insights as to how genes are regulated, functions of the encoded products of the genes, how gene products interact with each other in networks to affect biologic processes, and how we can use this information then that allows us new therapeutic strategies to treat diseases and new strategies to prevent diseases, both therapy and prevention for genetic and infectious diseases. So the first 2 speakers on our program will deal with 2 new areas ranging from the stem cell biology to very small RNAs, non-coding RNAs, microRNAs that have the ability to modulate gene expression in a very novel and important way. Then following a break, we'll have 3 speakers, Joel Rothman, Thomas Wimes, and Mike Mahan, that will share with us first aspects of apoptosis, programmed cell death, and how this has been what we've learned from studies of C. elegans in terms of the networks that are important and how that knowledge is relevant in understanding age-related mechanisms. And then Thomas and Michael will address 2 diseases, research, one a genetic disease, polycystic kidney disease, and then Mike Mahan and his recent research on vaccine developments for infectious diseases. Our first speaker is Dennis Clegg, and Dennis earned his bachelor's degree at Davis He then was a Jane Coffin Childs postdoctoral fellow at UC San Francisco, where he carried out work on nerve growth factor and the regulation of NGF expression and function that I think precipitated his interest in neurosciences. And in 1987 joined us here at UCSB, where he's carried out very important work on nervous system signal transduction and cell interactions in the nervous system. Dennis is presently professor and chair of the molecular biology department, for short, MCDB, as we heard earlier. He's also a professor in the biomolecular sciences and engineering program and a member of the Neurosciences Research Institute. And I mention these three affiliations because it's going to be a thread through the first session, and I think a real hallmark of how much of the research is done at Santa Barbara, an increasing amount of the cross or interdisciplinary activities between investigators in the life sciences and investigators in the physical and sciences and engineering, where we've come together to solve problems. And Dennis will summarize some of this information in the area of stem cell biology. His presentation is Interdisciplinary Research in Stem Cell Biology and Engineering. Dennis.
Thank you, Chuck, for that introduction. I'm going to start out by dropping the microphone and waking you up. Let's see, I'll let you do it. There we go. Start out with a picture, and ski season is still going on up in the Sierras since they have so much snow this year. There are three amazing things I wanted to tell you about this picture. The first is this downhill racer is going down the hill at about 65 miles per hour. The second is that this is a blind skier. And this is a picture of Mike May, actually from the town of Davis, California, who for many years held the speed record for blind ski racing. The third amazing thing, and perhaps the most amazing, is that Mike May can now see. And he was blind because he had spilled caustic solution on his cornea at age 3, and by surgery and corneal transplant using corneal stem cells, they were able to restore vision in one of his eyes. And I show this because to make the point that when we talk about stem cells and stem cell therapies that might be developed from stem cell research, they're already going on. And in fact, this type of surgery has been going on since 1999, and stem cell transplants are used in bone marrow transplants to treat leukemia. So, when we talk about the promise of stem cell research and we talk about developing tissues and cells for therapy, it may not be that far off, although there are still many challenges, especially in certain complex diseases. Now, this slide is from the NIH website, and it talks about the promise of stem cell research. And there is that promise where you can make all these different cells, bone marrow, nerve cells, heart cells, pancreatic islet cells, and treat diseases. But I wanted to point out two other areas that I think stem cells hold great promise in, too. The first, over here on the left, is drug development. And if we can generate these tissues in vitro, it may be possible to test new drugs on these tissues as a first step to develop new drugs and look at toxicity. And this could result in faster drug development. Those of you in the biotech industry know that making a new drug is a long road to hoe, and this may speed things up. The second thing has to do with developmental biology. We still don't understand how the process of development works. And by making these cells turn into things in vitro, we can recapitulate the developmental program and try and get at the genetic program that controls cell differentiation, and therefore increase our gain in knowledge in the area of development. Now, back to this middle area here, tissues and cells for therapy. Some things may happen soon, some things may take a while, but there are amazing types of experiments going on right now, and things are starting to work their way into the clinic. And I wanted to show you a little video clip from the work of Evan Snyder, who's down at the Burnham Institute and heads their stem cell program in La Jolla. And this has to do with spinal cord injury. And what they've done in this experiment, shown in the video here, is to induce a spinal cord injury to a rat, and you can see the rat can no longer get along very well. But then they tried injecting neural stem cells, adult neural stem cells from rat brain on a unique polymer scaffold. And here's what they see in rats injected with the stem cells 70 days post-injury. And they've gotten the same results, other researchers have gotten the same results using human embryonic stem cells. The mechanism of repair is not entirely clear, but they see dramatic types of recoveries, not complete, but still a lot better than the negative control. And so it's given great hope to people with diseases in spinal cord injury. I'm going to give you a little bit of an overview of what's going on here at UCSB, and then in the end talk a little bit about the research in my lab having to do with eye disease. And as previous speakers have mentioned, there's a great culture of interdisciplinary research here, and we form what we've been calling sort of a consortium for stem cell biology and engineering. Because if you think about stem cells, there's a lot of biology to work out, but there's also a lot of engineering in terms of how do you deliver cells to the right place, how do you sort cells, etc. And we have people that have gotten together and met and talked about possible directions in stem cell research. Actually, a meeting started by Ken Kosick right soon after the passage of Proposition 71 in 18 different departments and centers. And I put the type so small that if I forgot your name, you won't even know it. So the goals of this consortium are to understand the basic biology of embryonic and adult stem cell differentiation and function, to discover new methods for stem cell growth, sorting, and delivery, so those are the bioengineering types of problems. And then there's a particular focus on generation of ocular cells for the treatment of eye disease, following on the strong vision science research going on here at UCSB. As Martin mentioned, we do have funding from the California Institute for Regenerative Medicine, which of course was conceived with the passage of Prop 71 in 2004, and the proposition called for a bond issue that would fund $3 billion of research over 10 years. And as most of you probably know, the bond issue is still blocked by lawsuits. But the CRM was able to award the training grants using donated funds. And in fact, just last month, the Alameda Court did uphold the constitutionality of the stem cell program in its entirety. Unfortunately, the opponents are going to appeal, so this still may be tied up in the courts for another year. But the CRM is moving forward. They've raised additional funds now and want to have additional calls for grants, and it's a very exciting time for stem cell research in the state of California. So let me give you just a little overview of some of the projects that are funded by this training grant. First, there are a couple of projects having to do with adult stem cells. Todd Kippen is a new assistant professor in the psychology department, and he's interested in the mechanism and functional consequences of adult neural stem cells in the mammalian brain, working on mouse models. And there's increasing evidence that some psychiatric disorders may actually be related to abnormal stem cell activity, and he's exploring that. Steve Fisher in the MCDB department is interested in stem cell activation after retinal degeneration or during retinal degeneration, and he's found that some cells appear after retinal detachment that have stem cell properties. Looking at embryonic stem cells, you'll hear a little bit later from Ken Kosick, who's interested in the role of microRNAs in regulating differentiation of human embryonic stem cells. And then you'll also hear from Joel Rothman, who's interested in genes that specify stem cell competent states using worms, oops, wrong way, using worms as C. elegans as a model system to discover genes, and then transitioning to the human embryonic stem cell to see if conserved genes may have similar functions. And then likewise, Bill Smith is also using model organisms, and he uses the tunicate, the sea squirt, the Siona ascidian, to identify novel growth factors that regulate stem cell fate. And again, he'd like to transition that work into human embryonic stem cells. And both of these investigators are collaborating with Jamie Thompson, who is the director of the Y-Cell Institute at the University of Wisconsin. And Jamie's interested in setting up a satellite laboratory here in the CNSI building at UC Santa Barbara. Now, on the engineering side, Pat Daugherty's name came up earlier in collaborations with Erkki Ruoslahti. He's also interested in looking at stem cell surface epitope mapping to identify novel tags on stem cells and stem cell derivatives to allow screening and sorting using peptide strategies, using peptides expressed on bacterial surfaces. And then I'm involved in a project with Craig Hawker and Matt Terrell looking at synthetic and natural extracellular matrix regulation of stem cell fate. If you think about cells in the body, they live in a three-dimensional world, not in a dish, and it's likely that interactions with extracellular matrix may regulate their fate as they turn into different tissues. And then finally, Tom So, who's also collaborating with Jamie Thompson, is interested in developing new sorts of cell sorting technologies. And he's been working with, actually with Pat Daugherty and Carl Meinhardt on something called a DAX, a dielectrophoresis activated cell sorting. And in what is really exciting research, he's been able to develop a device, a small handheld disposable device that can sort rare cells with high efficiency based on the dielectrophoretic properties of either particles attached to cells or the cells themselves. And this could be very useful in stem cell research because you generate all these different cell types and you want to purify them to use to treat separate conditions. So finally, I wanted to talk a little bit about work going on in stem cells in ocular biology. And in a collaborative project between my lab and several other labs, we're interested in whether or not we can differentiate human embryonic stem cells to generate a cell type called the retinal pigment epithelial cell, or RPE cell. And this is actually a collaboration with a company as well, with Advanced Cell Technology, Mike West and Robert Lanza and Irina Klemenskaya, which has its headquarters in Worcester, Massachusetts, and also offices in Alameda. And why the RPE? And what is the RPE? Well, if you look at an eyeball, in the back of the eye, you find the retina, which is this white layer, and then behind the retina is a little black line, and that's the retinal pigment epithelial cells. And when you look into somebody's pupil and you see that it's black, that's because there's a layer of black pigmented cells behind the retina. And what they do is function to nourish and support the photoreceptors. So again, here's an eye. If we look at this little square here, this is from the Human Brain Coloring book where I learn all my anatomy. You can see these RPE cells here that extend apical microvilli that wrap around the photoreceptors and provide them with important nutrients and help them to function. And we're interested in RPE cells because of a specific eye disease, macular degeneration. And part of the work is going on in the Center for the Study of Macular Degeneration here in the Neuroscience Research Institute. Macular degeneration is the leading cause of blindness in people over the age of 55 in North America. And it's characterized by a progressive loss of fine acuity vision in the central portion of the visual field. So if you have this disease and you look out at something, you'll see a big black spot in the middle of your visual field. And it's particularly devastating disease for the elderly that want to sit and do knitting or watch TV or something like that. And it's very common. Something like 3 out of 10 people over the age of 75 have some signs of this disease. Now, we don't know entirely what the cause is, and there are no good treatments, But evidence is accumulating that the loss of vision is due to the death of RPE cells. And because the RPE cells die, the photoreceptors die. And there's increasing evidence from work by Link Johnson and Don Anderson and colleagues that this may be due to an autoimmune attack where there are certain variants of the complement factor H that are now associated with this disease. So if we could replace the RPE by some kind of cell-based therapy, maybe we could come up with a treatment for this disease. Now, I saw the guy in the back row scratching his head saying, what the heck is the macula? And so this is what the macula is. It's a tiny little part of the retina, a circular area about 1/10 inch in diameter, located just above the optic nerve here. So you can see, and that for some reason, unknown reason really, is the region where the RPE cells die and the photoreceptors perish as well. So our collaborators at ACT published a paper last year in cloning and stem cells where they reported that they could turn human embryonic stem cells into RPE cells. And what's shown in this picture here is a plate of human embryonic stem cells, the white cells here, that are put into conditions where they start differentiating into all kinds of different things. And right here, the arrow indicates a little colony of cells that has pigment. Well, they've looked carefully at these cells, and we've been helping with the analysis, and it turns out these cells have properties of normal RPE cells. And what I've shown in this picture here is work from Link Johnson's lab, where we show normal human RPE on the left and in vitro human stem cell-derived RPE on the right, stained for 3 different proteins that RPE normally express, and that's shown in green. And if we just look at the top panel, the red immunofluorescence, or the red fluorescence, is actually autofluorescence from lipofuscin, which are basically the lysosomes of these cells. They accumulate fluorescent compounds as they work in their regular life. You can see actually the human stem cell-derived RPE have less or very little of this lipofuscin, which may be a good thing because toxic compounds are thought to accumulate in this material. And the green in this top panel is a protein called emprin, and it's known to have a basolateral distribution in vivo, and you can see the same kind of basolateral distribution in these cells growing in monolayer. So we think that these HES-derived RPE look a lot like RPE cells in the markers that they express. We've also looked at the level of the electron microscope, and you can see apical microvilli here on these cells, as well as melanin granules, very typical of RPE cells. And all indications are that they are very similar to RPE cells. Now, one question that we've had is how stable are they? If we take these and grow these, will they throw off stem cells and start to generate tumors? How stable is the phenotype? And we're working on that question now. If we take an original culture here and take a clone of cells here, a colony of pigmented cells, we found that if we just pick this out and grow it as an explant, the cells will spread out and retain their pigmentation. Whereas if we take this and trypsinize it and dissociate the cells lose pigmentation. And so eventually they do start to recover the pigmentation. But this question of how stable the phenotype is something that we're working on right now. We're also interested in the steps that a cell takes to become an RPE cell. What is the gene expression regulation that goes on? And Sherry Hikita, working with Ken Kosick, has been working to identify microRNA expression signatures in developmental stages of RPE differentiation. And a number of microRNAs are predicted to target key transcription factors that are involved in differentiation. So let me wrap up and just tell you what I've said so far, given you a little overview of what's going on, and told you a little bit about our work to generate these RPE cells in culture. And we found that stem cells, human embryonic stem cells, a legal Bush-approved cell line, can be coaxed to differentiate into ocular cells. And the RPE displays many characteristics of native RPE. And our future research will address regulation of differentiation, functional abilities of these cells, and stability of the phenotype. And I think in the future, you'll see more of the types of operations that have restored vision in this skier. Thank you very much.
So our second speaker in the program is Ken Kosick, who is one of the newer faculty members that joined us here at Santa Barbara. Ken is a neuroscientist, and he will present some of his work on microRNAs. But first, a bit of background about Professor Kosick. He earned his initial degree in English, and then went on into medicine and earned his MD at Pennsylvania, and did an internship and residency in neurology. And I think it's at that stage that Ken saw both the clinical aspects of research, but the basic science aspects of research, and moved toward studies in neurosciences, where he has made very seminal contributions. After his training, he joined the faculty at Harvard as a professor of neurology and neuroscience, where he did very important work in many areas of neuroscience, but in particular on the protein tau that interacts with microtubules, a microtubule-associated protein, and also work in the area of Alzheimer's disease-related biochemical and mechanistic questions. He received several awards in recognition of this work: the Whitaker Health Science Award from MIT, the Denny Brown Award from the American Neurologic Association, and the Zenith Award from the Alzheimer's Association. We were fortunate to recruit Ken. He joined us as a faculty member here at UC Santa Barbara 2 years ago as the Harriman Chair of Neurosciences and as co-director of the Neurosciences Research Institute. His primary academic appointment is as a professor in molecular cell and developmental biology. And among Ken's research interests are the question of microRNAs, small RNAs that are non-coding RNAs that are very important in the regulation of gene expression, not only in developmental processes, but now, as we're learning, in certain disease states, in the interaction of pathogens with hosts. At any rate, Ken will present some of his recent work on microRNA research. The title of his presentation is "MicroRNAs, a Novel Class of Targets for Medical Intervention."
Thank you, John. Okay, well, there's always a slightly nervous moment when you have emailed your slides in ahead of the talk, and you don't know for sure if it's going to be your slides and what slides are going to show up. But I've had it said to me that a good speaker should be able to talk from anybody's slides. So we'll see what's here. And so good. This is it. And so as Chuck mentioned, I was very fortunate to be able to join the faculty here and to co-direct the Neuroscience Research Institute. And I've just listed a few of the things going on within the Institute. However, I want to say just one word that we're at an extraordinarily opportune moment on this campus from many, many perspectives, among which is that there is a kind of a an explosion of interests from many disciplines in the neurosciences, all the way from people like Frank Doyle doing basic computational work on circadian rhythms, all the way to the work that Mike Gazzaniga has brought here on fMRI imaging. And I think the connections that the Neuroscience Research Institute can build with these people is going to be really very positive for the future. Today I want to talk about microRNAs and how important, fundamental, revolutionary microRNAs are to biology and biomedicine. As Chuck mentioned, these are just 21 nucleotides. They don't code for a protein. There's just genes sitting there that sequences of DNA that make a small RNA of 21 nucleotides, and that's it. And then those nucleotides then go on to do some things that are truly amazing, and I'll talk about. But they have really turned out to be so fundamental that we really have to think of them to position them right down to where they fit in the central dogma of biology. Which I show here, which every high school student knows that DNA is made into RNA, is made into protein, and what the, let's see, this is a, is this going to light up somewhere? Yes. Okay, so I didn't want it to shut off the computer. What happens really is that you get a set of RNAs that are feeding back and regulating themselves. And that really may sound very, very simple, but it introduces a level of regulation here which was previously not appreciated at all. This was a whole field of biology that could probably have been discovered 30 or 40 years ago, but because they're so small, they had been missed and discovered only about 5 years ago, and with their discovery has now opened up many different aspects of biology, and I'm going to just talk about one of them. The way to think about the importance of this layer of regulation is that you're familiar that in a bacteria we have this the DNA sitting here exposed to the entire cell, and then this amazing evolutionary step happened in which cells became what are called eukaryotes. They formed nuclei, and now the DNA is housed here within the nucleus. It's kept separate from the whole rest of the cell. A kind of compartmentation has taken place. And we know that there are, that one of the complex layers of regulation are called transcription factors. They're encoded in the genome. They make an RNA and a protein, and the protein comes out to the cytoplasm and back into the nucleus, and those transcription factors will combinatorially bind, and there could be up to 1,000 of them, combinatorially bind to the front end of genes and in various combinations regulate their expression. So this is really key. This is why so many transcription factors are looked at as potential targets for cancer and other diseases, because they are up until now, they are the main layer of gene regulation that we know about. Along come microRNAs, and now with microRNAs we have another layer of regulation that is perhaps as complex or almost as complex as the transcription factors. So DNA in the nucleus makes RNA, the RNA moves out now into the cytoplasm, and out in this region, whether or not that RNA is going to get translated into a protein is being controlled by the microRNAs that also have many, many components. There are now known in the human genome to be about 350 of them, and they also bind combinatorially in very interesting ways to regulate large numbers of messenger RNAs sitting out here and determine whether or not a protein is going to be made. So, an entire novel layer of regulation. Here's what a microRNA looks like. It is just these shaded 21 nucleotides, and it comes from a precursor of 70 nucleotides that forms a stem loop structure. And here's the way a microRNA looks when it actually binds onto a messenger RNA. This happens to be an example I've taken from the work of Gary Ruvkin in using some C. elegans genes, but this is true in humans too, where microRNAs, these short 21 nucleotide transcripts, bind onto a messenger RNA in the black line here. You see here's the poly A, the cap, and it binds on and will prevent its translation. Now, the holy grail in the microRNA field is to understand which microRNAs bind to which messages. And you might think that's a simple challenge because we have this Watson-Crick base pairing, and you should be able just to predict that A's and T's hook up and C's and G's hook up, but it's not so simple because the way microRNAs bind is that sometimes they're inexact matches, and sometimes they match completely, but sometimes they loop out. So simply blasting the genome with the microRNAs will not give you the answer. And therefore, it's going to be very important for us to devise techniques to find targets in a very rapid way. It's something that we're working on very, very assiduously. So, and one last word of introduction here. The whole microRNA biogenesis pathway is now more or less understood. As I mentioned, microRNAs are also genes. They're encoded in genes. They get their precursors are in the nucleus, these stem loop structures. They get moved into the cytoplasm, and then there's a series of enzymes that come along, cut out just the strand that's going to bind the message and sit on the message here to either cleave the RNA, the double-stranded piece of the RNA now, the microRNA hybridizing to the message, that double-stranded piece will become a target for an enzyme that likes that kind of structure and will cleave it and destroy the message, or it'll just sit there and inhibit translation in a way that we don't fully yet understand. And both of these can occur. This tends to occur a little bit more in plants, but they both occur with various balances. Okay, so where we came along in this field was to develop approaches to try to understand microRNA expression on a large system scale. And we started this work with with profiling just 180 microRNAs on a nylon array and looking at microRNA expression patterns in a number of different conditions. And you heard Dennis mention something about stem cells, which we're interested in, and neuronal differentiation. There's a lot of areas that we could talk about today, but I'm going to mainly focus on brain tumors, because one of our hypotheses about microRNAs is that they are very critical for both determining and maintaining cell identity. So the areas where both we and other people think that microRNAs will have a very important role is one setting, as you heard from Dennis, that where a cell has to find its identity. That's what a stem cell is all about. But another setting where microRNAs are perhaps equally important is in cancer, where a cell loses its identity. And these are sort of opposites of each other, and microRNAs already has been clearly demonstrated to have important roles in both. So just to introduce this, I'll show you some collaborative work that we did. Here's some of the profiling that was done by a postdoc in my lab named Anna Krachevsky. And I'm just going to say a word or two about something I know very little about, going outside the nervous system now. It's a little dangerous for me to do that. So I'm going to say a word or two about the hematopoietic system here, just to introduce the topic. But in collaboration with Klaus Rejewski and Jana Rao, we looked at profiles of a number of different cells in hematologic lineages and compared it to those in neurons. And basically, if it's white, it's a white dot, it's along here are all the different microRNAs. So a white square means it's very abundant, and a brown square means it's not much there at all. And there are a bunch of microRNAs here that are mostly neuronal, and a bunch here that are mostly present in these hematopoietic cells that are differentiating along different lineages shown here. And here are cells that probably people in the audience are more familiar with than I am, B cells, T cells, mast cells. And the point of this slide, the whole point, is right here at these nodes, when cells are changing their identity, that is, you can see that of the 330 microRNAs, there's really a relatively small number that will be up and down regulated, because a microRNA is able, by binding to maybe 200-300 different messages, it's an upstream control element that can control a broad layer of mRNAs. And in so doing, with these small changes, can direct cells into these various lineages. So this is sort of gets more directly as to what their functions may be. And we're now doing exactly this again, looking at neural cell lineages. Okay, so now to bring you exactly on the subject at hand today, I want to talk to you about specifically the microRNAs. In brain tumors, I'm going to talk about glioblastoma is a deadly, deadly tumor once it's diagnosed with about a 1-year life expectancy in humans, especially the most grave form, the glioblastoma multiforme, and how microRNAs will have a role in these tumors as an anti-apoptotic factor. So here's just a little something I already mentioned, how serious these tumors are with this very short survival time and are completely inadequate therapies at the moment. So using the array system, we looked at a bunch of GBMs, or glioblastoma multiformes, and found that this dot right there, which corresponded to microRNA 21, was really consistently increased in many of the arrays. And then going to northern blots, we also saw down here, you can see, right here, you can see that the MIR21, microRNA 21, was increased as opposed to a number of different controls. And here in these glioma tumor cell lines, it was also very increased. And we did a lot of controls because for any kind of work with tumors, you always have the problem of the control. If it's called a glioma, but what's the cell of origin? Is it a neuron? Is it a glial cell? Is it a stem cell sitting in a niche somewhere? You don't always know what the control is. So you have to do lots of controls. And once you do that, you see that this is increased quite specifically in the malignant tumors. It's turned out subsequently, I'm not going to get into this, but that MIR21 is also increased in other types of malignancies, but it's really exceptionally high in the glioblastomas. And here's the point of really, this is one of the central points I want to make, is that there are techniques in which you can actually deliver what you might call an antisense to the microRNA. And what I mean by that is you take the complementary sequence, do a little bit of chemistry, and you can now have a sort of an anti-MIR21 bind to MIR21 by synthesizing the complementary nucleotides, introduce it into a cell, and suppress it. So we did that, and the chemistry involved here, they're called locked nucleotides. Another type of chemistry are called 2O-methyls. We don't have to get into that. But basically what we saw was when we inhibited MIR21, we were able to markedly decrease the cell number here in this, when we use 2O-methyls or the locked ones, but not in any of the controls. Okay, so here in culture, cell number has gone down. Is that because the cells have, we've slowed down cell division, or we've increased cell death? And the answer is that we were increasing cell death. That and this is shown here. You can see this almost looks like the inverse of this, because these levels here with the treatment, those enzymes that induce apoptosis or cell death, the major executioners in the apoptotic pathway, caspase 3 and 7, are markedly increased in the presence of the inhibition of MIR21. So we believe that that something about a glioma will cause an elevation of MIR21 that then begins to inhibit many genes in the apoptotic pathway, and therefore the normal cell death that should be occurring is not occurring, and the tumor malignancy very much increases. So, in conclusion then, we believe that miR-21 is strongly overexpressed in high-grade gliomas. This work has now gone far beyond just what I'm showing here now, that both ourselves and other people, there's one of the scientists also in Boston named Todd Golub, has examined many different brain tumors, and we now know that it's possible actually to look at microRNA profiles as actually diagnostic and prognostic for brain tumors. So the whole profile is telling us quite a bit. We know miR-21 inhibition will trigger the activation of some apoptosis, and that the aberrantly expressed miR-21 is contributing to the malignancy. So this has given rise to a word that we're beginning to hear more frequently, called oncomiRs. These are miRs, microRNAs that may be involved in tumorigenesis. miR-21 is not the only one. And it gives rise to a lot of ideas about how we can really think about critical targets in cancer. So again, talking and just showing a specific example here, there's dysregulated miR-21. This means that target mRNAs, and this miR-21 will have maybe, as I say, 200 or 300 targets. Being able to identify those targets will greatly expand the repertoire for intervention in cellular processes, and opens the way to discovery to multiple targets in carcinogenesis. Because, but it may not be desirable to actually use all of these targets when we can cover all of them with one nice, discrete, strategic surgical intervention, getting at just those 21 nucleotides that are dysregulating the system. So the last thing I want to show is is to bring you right up to date and show you what's going on in the lab now, which is this work here is in collaboration with a group at Applied Biosystems that has discovered a way to use real-time PCR and multiplex this to analyze all 330 microRNAs, no longer on a nylon filter in which you need a fair amount of RNA and the radioactivity, and it's just a little bit more messy, but to really analyze even more micros, all of them, in picogram amounts in a test tube. And what they do basically is made primers to all the known microRNAs and go through a series of reactions here to actually give quantitative information for every single microRNA. And we're working with them on this now to analyze a number of different samples. We have data from a number of glioma lines and other conditions that I showed on that slide. And these large data sets are going to be really worth mining for an awful lot of information about targets. And here's basically what we do. We have the ability now to grow neurons in culture, laser capture discrete cells, and profile just a single or a handful of cells that are stained with a single antibody. So we can apply this to the stem cells if we want to look at specific stem cells that are stained in a certain way, or certain tumor cells. The surgeon can take a tumor out from the OR. The pathologist would look at it under the microscope and say, I'm worried about those few cells there. Can you capture them by a laser and give me a microRNA profile? And that now will have some, we already know, will have some diagnostic and prognostic abilities. And when you get pictures like this, you know, where you see all the microRNAs here and their patterns of expression here, like that. So, okay, so I'm going to just give the credits now for this work. The people in my lab are all listed here. We've got a really great group working here now in Santa Barbara. The people that did some of the tumor work before I came here were Anna Krachevsky, Jennifer Chan, and Sasha Levy was actually working with me here but has now moved on to NYU, and a number of collaborators listed here have helped with the work. Thank you.
Joel Rothman, who will present the first talk, received his undergraduate training at UC Davis. He then earned his PhD at Oregon, where he studied yeast genetics and molecular biology, and then moved to, at that time, really breaking model organism, C. elegans, worms, to study embryonic development and from a genetic angle in the C. elegans system as a Helen Hay Whitney Fellow and an Embo Fellow at Cambridge. And after that postdoctoral work, then moved back to the U.S., but not quite to the West Coast yet, for a stop at Madison in the Department of Biochemistry. And then he joined us here in 1996 at Santa Barbara, where he continued his C. elegans research on developmental questions, and rapidly rose from an assistant professor in 96 to a full professor now. Joel is a member of the MCDB department. He's also a faculty member in BMSE and a member of the Neurosciences Research Institute. So yet another of our speakers that have broad research affiliations on campus. And the title of Joel's presentation is Death and Renewal, Discovery of Genes that Control Apoptosis and Developmental Reprogramming. Joel?
Great, thank you. Well, thank you. It's an honor to be presenting to you today. And actually, the title, I think, in the program is Aging and Age-Related Disease. And really, this is all about aging and age-related disease. We humans are actually some of the longest-lived creatures on this planet. That's not enough for most of us, I think. I think if I took a survey, most of you would agree with that. And I believe now from studies in a lot of model organisms, such as C. elegans, mice, and other creatures, that we are going to be able to affect a very significant increase in our lifespan. And I think it's certainly biologically tenable to think of us as ultimately becoming immortal. And I think the reason that we could become immortal is because of the special feature of mammalian biology, certainly that humans use a lot, and that is the feature of renewal, the fact that we replace essentially all of our cells in a constant way. So this notion of replacement is greatly illustrated by the fact that of our 10 to the 14th or so cells, about 100 billion commit suicide every day. And of course, another 100 billion are born. We're constantly renewing our cells. In fact, in about a year, your mass in cells is committing suicide. Your complete mass in 1 year is committing suicide and then being replaced by this renewal process. So I want to talk to you about our studies in understanding the process of death, in particular programmed cell death or apoptosis. I won't be talking so much about our other studies related to the aging process directly, although these are all aging relevant issues, and also renewal, and I'll be talking about how we're trying to understand the mechanisms one can use to reprogram the multipotentiality of cells, that is to create cells that, although initially were differentiated, become multipotential and could be used to renew tissues once again. So let me start by talking about this process of programmed cell death that is so widespread among mammalian biology and in humans. Programmed cell death, also called apoptosis, is a very striking process. It's a carefully orchestrated, choreographed process of cell suicide in which a cell is neatly disassembled in a way that prevents the cell from lysing and producing inflammation. And this process of apoptosis is critically important not only for forming who we were to begin with. In fact, our nervous system, for example, critically depended on sculpting events that require apoptosis, that involve apoptosis, but it's also a critical protective mechanism against disease. And so there are many, many diseases that are the result of dysregulated apoptosis. And let me just give you a flavor for some of those. First of all, apoptosis is critical for our survival. Were it not for our ability for our cells' ability to commit suicide, we, paradoxically, couldn't live very long. And so, for example, if apoptosis does not occur when it should have occurred, that can result in a large range of cancers. In fact, probably all cancers have underlying them the inability to commit suicide, the cells to commit suicide when they should have. And there are a number of other diseases that are a result of inactive apoptosis. Likewise, it is also true that apoptosis can occur inappropriately and cause disease. So all of our cells are loaded up with the machinery to commit suicide. All of our cells are on the knife's edge, and that machinery can go awry. And in fact, when excess apoptosis occurs, there's a large number of diseases that can arise. Here's some of them. Most, perhaps all, of the neurodegenerative diseases are the result of the inappropriate activation of the cell death program. Diseases like AIDS, and also, in fact, most of the damage that results in stroke and heart attack results from this programmed cell suicide process. So obviously, the cell death is one of the critical processes underlying many diseases. And we're studying this process of apoptosis using a really powerful discovery system, C. elegans. And this is a powerful discovery system not only for discovering new processes. So for example, Ken Kosick had mentioned to you microRNAs. MicroRNAs were discovered 15 years ago in C. elegans, and we've since discovered, of course, that they're used in, throughout the animal kingdom. The key regulators of apoptosis, caspases, were discovered in C. elegans, and Bob Horvitz won a Nobel Prize for that in 2002. But this is also a very powerful discovery tool for discovering not only gene functions, but in fact entire pathways. So we're approaching the discovery of pathways underlying these processes of cell death and cell renewal by understanding complete pathways and hoping to intervene in those pathways in the context of therapeutics and other medical approaches. Chuck had mentioned that this is the organism for which we have a complete description of the anatomy and development at single-cell resolution. But I think the characteristic of this organism that's making it extremely exciting to work with for understanding and describing pathways is the ability to knock out essentially all of its genes in a very easy way using this process of RNAi, which was in fact discovered using C. elegans. Now, of course, understanding these pathways would be useful only if C. elegans uses the same pathways that humans do. And in fact, it's very clear, and was clear from the beginnings of the sequencing of this organism, that the toolkit used to control most processes, such as cell renewal and cell death, are conserved between C. elegans and humans. Okay, so this just illustrates the cell lineage of this animal going from a fertilized egg to a 1,000-cell adult. We have a complete description of all of the cell divisions. And without looking at any details here, one of the salient features of this is if we zero in on a part of the nervous system, for example, you can see a lot of cells indicated by these red dots that commit suicide shortly after they're born. All of those cells die within 20 minutes or so of their birth. In fact, about 12% of all the cells of this animal commit suicide by this choreographed process of programmed cell death. And a cell suicide is a cell that looks like that. That's, oops, the arrow slipped, I guess. Those two cells there are each cells that have committed suicide by this process of cell death. So it's very easily scored. It's also readily amenable to genetic analysis. And the genetic analysis that we've taken on is to approach the architecture, to understand the architecture of the pathways controlling this delicate balance between life and death by using this process of RNAi. So we can do very large-scale functional genomics screens in C. elegans in a very straightforward way, and that is as follows. You can induce RNAi, that is RNA-mediated inactivation of messenger RNAs in C. elegans simply by feeding them bacteria that express double-stranded RNA for a particular gene. And what this means is we have available now in our freezer from Julie Ehringer's lab in Cambridge a complete library of the whole genome, all the genes in C. elegans represented as bacterial clones expressing double-stranded RNAs. And all we have to do is feed worms. This is remarkable, but it works. We simply feed worms on those E. coli bacteria clones. And simply the act of eating that bacteria containing the double-stranded RNA will induce RNAi for that specific gene, will essentially eliminate the function or greatly decrease the function of the encoded gene product. And so what this means is we can screen effectively, or most of the entire genome of this animal, simply by taking worms, feeding them on bacteria expressing the appropriate double-stranded RNA, and scoring their progeny in the next generation a few days later. So this provides a very rapid system for discovering new gene functions. And we've applied this to understanding the control of cell death. We've identified, just in the last couple of years, over 100 new programmed cell death regulators using this system. Most of these regulators are proteins that prevent programmed cell death from occurring. We've also started to identify some new components that are required for activating cell death. as well. And I obviously don't have time to describe those 100 genes, but I want to give you a little flavor of how we can take apart this information and look at the architecture of how they function. So first of all, this just sort of represents the kinds of things we can see. This is a normal C. elegans embryo with a cell death right there. Here's one that's been activated for a gene we discovered, ICD1, inhibitor of cell death 1. You can see these embryos fill up with apoptotic corpses. So cells are dying willy-nilly all over the place. In fact, we find that this is really effectively a severe neurodegenerative disease in this animal. We see tremendous loss of nervous system cells as well as other cells. And so using those kinds of phenotypes and others, we can come up with a description or a set of proteins that are involved in suppression of cell death. This is a summary of one particular analysis that Pradeep Joshi, who's sitting in the back there, postdoc in my lab, did to identify genes that are involved in suppressing the death of cells in the germline. And what, of course, immediately upon seeing a phenotype like I described to you, we immediately know what the encoded protein is. We can go right back to the database and figure out what gene it is that led to that phenotype. And what we see is a range of protein types that, when inactivated, result in inappropriate programmed cell death. And one attractive feature of these proteins that Pradeep has identified is that a number of them are enzymes. That is, they're potential druggable targets. We could, in fact, in many cases, there exist inhibitors already that have been identified for these. So we can certainly already engage in a possible search for existing inhibitors that may be useful for activating the cell death program, for example, in cancer cells in which the cell death program had been suppressed. In addition, the genes we identify, a number of them have already been associated with neurodegenerative diseases. So again, these are proteins that when you inactivate them, result in inappropriate cell death in C. elegans. We know that the human equivalents of at least these three are associated with neurodegenerative disease, although those neurodegenerative diseases had not been known to be the result of de-repressed apoptosis. So I think the studies in worms are telling us about the mechanisms of these diseases. And in addition, as I said, apoptosis is a first line of defense against tumorigenesis. And so you might imagine if these proteins are inappropriately activated, expressed, for example, at too high a level, you might inappropriately turn off the cell death program, and that could lead to tumorigenesis. And in fact, a number of the proteins we've identified in our screen are associated with cancer. All of these things are upregulated in various kinds of human cancers indicated here. So although none of these had been known to be associated with apoptosis, they are associated, they're overexpressed in these tumor types. Certainly, one can imagine that, in fact, in each case, perhaps the reason these genes are contributing to tumorigenesis is they're inappropriately turning off the apoptotic program. Now, what we'd like to do is take a list of genes, over 100 genes, that we know are functionally relevant in this process, and try to understand the architecture through which they act. That is, what is the network through which these genes function to control this critical switch? And we've embarked on an analysis of this architecture of a network, partly in a collaboration with Ambush Singh's lab in computer science. Again, this illustrates the importance of the cross-fertilization on this campus. Ambush's lab has worked closely with ours, developing algorithms and methods for essentially data mining, integrating data sources from many different locations, and looking for the probabilistic affinity of any two proteins for one another. I'll explain that better in a second. So basically what we do is we dip into as many database sources as we can get, microarray expression database sources, literature sources, location in the genome, interactome data, many other sources, and look for a clustering of activity, that is, behavior of any two proteins across many of these databases together, to develop what we call a likelihood ratio for each pair of proteins. And all that likelihood ratio means is how likely is it that those two proteins may function together in the same process. Okay? So what we're really doing is taking all the database information out there and looking for the probability that any two proteins work together. And this has been enormously productive. One of the networks that we've worked up, I'm not showing, this is a network that was actually used, that Pradeep developed using a different algorithm recently published by Zhang and Sternberg. And in this case, Pradeep is taking 20 proteins that he's identified as being critical for regulating apoptosis in the C. elegans germline. and he's looking for the association of these proteins with one another based on this database mining approach. And so what's shown in this complicated slide is one remarkable observation that jumped right out at us, and that is a remarkable connectivity between these proteins. So these numbers indicate how related these proteins are to one another. Basically, for example, if he takes this protein and says, okay, let's use this algorithm and say from 1 to 10,000 going across the genome, which protein has the highest likelihood of functioning with this, which has the second highest, and so forth. What he did is he cut all of for each one of these, he developed a list, and he cut that list off at 100. So he took the top 100 out of 10,000 for each one of these. And the remarkable thing is that every one of these proteins is on the list in the top 100 of at least one other protein from that set. So for example, this protein right here is 6th. It is the 6th most likely protein in the entire genome, or at least of 10,000 proteins, to function with this protein. That's what that 6 means. And likewise, the other way around, this guy is 13th on the list of this guy's set. Now, those are extremely high likelihoods. We were amazed that, in fact, the entire set of proteins that Pradeep identified, simply by functional assays using these screens, are all interconnected. There's a huge amount of connectivity, and in fact, all of these links are in the top 1 percentile. In fact, the average percentile overall is the top 0.4 percentile, or the top 99.6 percentile, depending on if you're taking the SAT or getting a grant, I guess. Okay, I don't know why they flip those around. So the point is that there's an enormous connectivity. The computer here had predicted the result that Pradeep got in the lab. The computer would have predicted these results. So this is telling us about the relationships between proteins from the screen. It's actually going to shortcut a lot of experimentation for us. But it also allows us to look for pathways. And so from these kinds of analysis, Pradeep is able to see, by taking this information and melding it with phenotype information, he sees a cluster of proteins that seem to function fairly specifically in the cell death process. Other proteins from his screen that have a relationship to cell death, but as well seem to be involved in cell proliferation, and another protein that is involved in cell growth or limiting cell size. So there are different pathways that are emerging from this, and to discover complete pathways is very useful if you're thinking about drug design. We believe these represent modules of protein activity. I think one of the revolutions in drug design that is coming forward, and will be more so in the future, is developing drugs that are cocktails of multiple drugs. That is, some combination of drugs, especially already approved drugs, if you can put them together in combination so that you shorten the approval time a lot, you may well be able to develop complete new therapies from extant drugs by hitting multiple targets simultaneously. And these pathways will allow us to infer which targets one needs to hit. Notice these processes of cell death, cell proliferation, and cell size, or cell growth, are all processes that are aberrant in cancer. And in fact, within this set of genes are proteins, these 4 proteins here, that are directly tied to cancer. And some of the ones I mentioned to you, as well P53, probably the most important tumor suppressor in human cancers. Okay, so this pathway approach is very exciting to us, and we're very struck by the fact that the proteins that we identify using this functional genomics screen seem to be highly likely to function in the same process based on what the computer tells us. Okay, if I could just take a couple more minutes and look at the other side of the coin from death, this issue of renewal. We're using this gene discovery system as well to look at systems that we might be able to develop to generate new kinds of stem cells. And so what we're looking at is how can we program multipotentiality of cells. Okay, so this summarizes the problem. So stem cells, which have been introduced very nicely by Dennis Clegg, and Ken talked about it a bit. Stem cells are self-renewing cells that produce more stem cells and also produce various kinds of differentiated cell types. And that process we think of always as occurring in one direction. You can go this way, just like the central dogma of biology. You can go this way. You never go backwards. You make a differentiated cell, you're locked there. If you could go backwards, then actually the politics of stem cell biology wouldn't be a big problem, right? In fact, is it possible? Can we reverse this process? Can we take a differentiated cell and turn it back into a stem cell. I would argue there is absolutely no biological reason that that's untenable. We already know you can take the nucleus from a differentiated cell and turn it into an entire organism. So of course, at the level of a nucleus, this is a completely reversible process. Can we do it at the level of individual cells? So we're using the C. elegans gene discovery system to look for the steps that one might interfere with to be able to reverse this. Imagine if you could take a cheek cell or some other differentiated cell type from a patient, do some sort of hocus pocus to it, a combination of RNAi and drug treatments, cause that cell to go back to being a stem cell, and now you have a stem cell created in a patient from differentiated tissue. You can get as much as you want, as much of that as you want, and this will work regardless of who's in the White House. So, all right, so here's our strategy. We are able to reprogram the development of all the cells in the C. elegans embryo. So we've studied, for example, a lot of the genetic circuitry, regulatory circuitry that triggers the formation of the intestine. We have a set of proteins we've identified. These are called end proteins for endoderm determining proteins. And we can turn those proteins on throughout the entire embryo. When we do that, we can turn a C. elegans embryo, a whole worm essentially, into a whole C. elegans intestine. That's it. So this is an embryo, but every single cell in that embryo is a gut cell, and that's all there is. Okay? So we're able to reprogram all the cells that would not become intestine into becoming intestine. Now, the interesting thing about that is that there is a window of time in which we can do that. So this represents that window of time. If we do that experiment, if we express the end proteins throughout the entire embryo at a critically late time in development, after this window of time, we can no longer get that conversion of a precursor cell into a gut cell. Those cells have locked into their differentiated cell states. And so the multipotentiality of cells in this embryo is dropping off as we hit about 2 hours, about the time we know various cell fates are being specified. What are the mechanisms that shut off that multipotentiality, and can we reverse them? Can we eliminate, or can we open up the multipotentiality, extending it, and thereby perhaps reverse the differentiated state of cells? The answer appears to be yes, based on experiments in my lab now. So we've done an RNAi screen, essentially to look for genes that, when inactivated, extend this window. And we get a number of genes that can do that. Here's an example of one gene. This is a worm inside an eggshell. And this area right here is expressing an intestine-specific protein. It's a transcription factor that normally is only in the intestine, but it's occurring throughout the region normally occupied by the nervous system. And that is because we switched those cells over very late in development because we inactivated a particular type of transcription factor called Lin26. So this just shows you that's Lin26 is one of these other proteins that, when inactivated by RNAi, extend that window of competency to be reprogrammed into this endoderm or gut pathway of development. What we've achieved then is, at a very late time in development, when these embryonic cells have already become committed to specific cell types, we've been able to take what would be differentiated cells and convert them over to another cell type, at least by virtue of the fact that they're expressing a marker of the intestine. So we're opening up this window. It looks like we're creating new multipotentiality of cells later in development. Well, we've just started this. This is very promising, and we can, I think, tackle the entire genome using this RNAi screening approach that I've mentioned to you. The proteins that we've identified that restrict multipotentiality so far are a number of transcription factors, also proteins involved in chromatin structure and chromosome regulating factors, and also one protein that appears to be a serine protease, in fact, a potential druggable target. This is just the beginnings. We think, continuing in this way, we should be able to get the entire universe of proteins that, when inactivated, cause a cell to back up in its differentiated trajectory and be reprogrammed into other cell types. Okay. So let me just summarize what I've told you. We, I think, have very powerful methods for discovering pathways in this system. We've discovered a large set of new apoptotic regulators that can point to new diagnostic and therapeutic targets in cancer and neurodegeneration, and perhaps we'd like to see how those pathways segregate out so we could even propose possible cocktails that could be used to control these diseases. And our work on developmental programming may provide us a means for engineering novel multi-potential stem cells, because of course we'd like to apply this information to mammalian tissue culture cells and see if we can likewise reverse their differentiation. And I need to thank the people in my group involved in these projects. And I guess I won't go through all of them, but a number of people involved in the cell death work, multipotentiality and so forth, and other projects in the lab, and the collaboration with Ambush Singh's lab working on this network computation. I have to thank Julie Ehringer for providing this extremely valuable resource, the RNAi library, that we use daily, and the institutes that have supported our research. Thanks a lot for your attention.
Next presentation is from Thomas Wimes, our newest member of the faculty in the MCDB department. Thomas received his undergraduate training in Germany and actually his PhD as well at the University of Cologne. He then moved to UCSF as a Lenin postdoctoral fellow supported by the Humboldt Foundation, where he initiated studies that led him to subsequently join the Cleveland Clinic as an investigator and an assistant professor at Case Western. And we were fortunate to recruit him to our department in UCSB last summer. Thomas has research interests in a couple of areas. One that's immensely important that we'll learn about from him has to do with a genetic disease, polycystic kidney disease that can lead to renal failure. And at this point, as far as I understand, there is really no preventative treatment for this disease. As I said, it's a genetic disease. Mutations have been identified in a gene that encodes a protein, polycystin 1, that Thomas has made really very seminal advances in understanding how this functions to, as we've heard in earlier talks, alter signaling by altering the protein-protein interactions that occur. And in this case, interactions are altered to activate signaling, and he now has shown with an inhibitor that you can perturb this to reverse some of the consequences. And we'll hear about these studies in his talk that we lost our summary title slides, but the title of the talk is New Functions of Polycystin 1, the Protein Affected in Polycystic Kidney Disease. Thomas.
All right, great. Thank you very much, Chuck, for this nice introduction. I'd like to tell you today about a project in the lab that is one of a couple of projects, so this deals, as Chuck just mentioned, with polycystic kidney disease. Most of us had probably never heard about polycystic kidney disease. It's not one of the two well-known diseases, unjustly so, because it is actually the most common life-threatening genetic disease. It affects about somewhat more than 600,000 Americans. And interestingly enough, it affects more people than cystic fibrosis, muscular dystrophy, hemophilia, Down syndrome, and sickle cell anemia combined. It is one of the leading causes of kidney failure in the US, and two genes have been identified. If either of them is mutated, it leads to the disease. They're called PKD1 and PKD2. They encode for the proteins called polycystin 1 and polycystin 2. Polycystin 1 accounts for the vast majority of the cases. The functions of those two proteins, however, have remained largely unknown, which has led to difficulties in trying to devise a strategy to treat those diseases because we don't know what essentially is going wrong. And we've been focusing on trying to identify the function of polycystin 1. First let me just show you some of the clinical features of ADPKD. As the name suggests, epithelial-lined cysts in the kidney are the main phenotype. There can also be cysts in other organs. There can be aneurysms in the brain, hypertension. But the biggest problem for patients is that typically by the age of 50 on average, about half of them will experience renal failure, which means that for their continued survival, they will need lifelong dialysis or kidney transplantation. There's currently no treatment available that would slow the progression of the disease or prevent it. This is how those polycystic kidneys in the end stage look like. As you can tell, they are several times the size of a normal kidney, which would be around this size here. They're very highly cystic, and those would be end-stage kidneys that would have probably stopped functioning several years already before this picture was taken, before they had been taken out of the patient. This patient most likely was on dialysis for a while already. At this stage, also there would be virtually no more normal renal kidney tissue left in those organs. So how do those cysts form? They're thought to form from the normal renal tubules. As you know, the kidney is made up of renal tubules that are themselves made up of epithelial cells that line the central lumen here. It is thought that a single cell acquires a second hit somatic mutation, which allows it to proliferate faster, form a small cyst, which eventually goes larger and larger, disconnects itself from the nephron, and essentially grows in size over time. So we have been interested in identifying some aspects of the function of polycystin 1, which is shown here. It is a protein, a membrane protein that has a very large extracytoplasmic domain here, several transmembrane domains, and a fairly small cytoplasmic tail. And we have found that the cytoplasmic tail regulates two intracellular signaling events. One of them involves a protein called tuberin and another one called mTOR, which is what I will be telling you about today. The other event involves a transcription factor called STAT6 and another one called P100, which I won't have time to go into. So initially, one of the series of experiments that we made was that we focused on the cytoplasmic tail of polycystin 1, which is shown here. Again, here's the transmembrane domains, here's the extracytoplasmic domain. And we made fusion proteins where either the full-length tail or certain portions of the tail are fused to a transmembrane domain and another extracytoplasmic domain of a different protein. We made an interesting observation when we expressed this protein here, which we call NTM-PC1. So it has the N-terminal part of the C-terminal tail of polycystin 1 in a membrane-anchored version. That's what the M stands for. We express this in a cell line called MDCK. They're a very well characterized renal epithelial cell line. And we express this NTM-PC1 protein in them using a doxycycline-inducible promoter. So we can control the expression by either adding doxycycline or omitting it. And what we noticed was that expressing NTM-PC1 results in cell death. So we have a lot less cells here after a certain time point. And if we look at the cells, we see the typical signs of apoptosis. We see the condensed fragmented nuclei here. We can see DNA laddering on a gel. And if we take a further look by flow cytometry, we observed that at an early time point, 24 hours after induction of expression, we see a G1 cell cycle arrest. This is followed later by apoptosis. So what we saw was expression of NTM causes G1 cell cycle arrest followed by apoptosis. So we took a look to see what could be the mechanism and what other players might be involved. One observation is that NTM-PC1 localizes to a region that overlaps very extensively with a Golgi marker. So for some reason, this protein therefore localizes to the Golgi apparatus. And then we looked at certain candidate proteins. And for one of them, I'll have to introduce you to another disease first. A disease by the name of Tuberous Sclerosis Complex, or TSC, which is also a genetic disease that is about 10 times less frequent than polycystic kidney disease. It affects about 50,000 Americans. It is characterized by tumors throughout the body in many different organs, including the skin, kidney, brain. And there have been two genes identified that cause TSC. They're called TSC1 and TSC2. The proteins that they encode are called hamartin and tuberin. And interestingly, TSC patients also have kidney cysts, so this has been known for a long time. So for a couple of other reasons, we hypothesized that the TSC2 protein could have a functional relation to the polycystin protein. And those are that the two genes for the TSC2 and the PKD1 genes are actually immediately adjacent to each other on the chromosome, separated only by about 60 nucleotides, and this is very highly conserved in evolution. And therefore, there is a group of patients that have both of those genes affected. And in those patients, one observes a very severe early onset polycystic kidney disease, much earlier than if either one of those genes is affected singly. Furthermore, there was a report a few years back now that suggested that tuberin is required for the trafficking of polycystin 1. So we hypothesized that tuberin, the protein of the TSC2 gene, the tuberin might be functionally linked to polycystin 1. And we took a look at where tuberin localizes in MDCK cells. So it has this sort of punctate cytoplasmic localization here. Does not co-localize with a Golgi marker. However, if we express anti-MPC1 in those cells, now tuberin targets very prominently to the Golgi. And we can also show that it co-localizes there. Here's tuberin, here's this anti-MPC1, while in controlled cells, tuberin does not show this Golgi localization. So this suggested to us that tuberin might interact with this part of the cytoplasmic tail of polycystin 1. And to test this biochemically, this here shows a co-immunoprecipitation experiment where we now in this case express the full-length tail of polycystin 1 or the C-terminal part of the tail that's missing this interacting region here. And what we noticed was that the full-length construct can pull down tuberin. The one that is missing this interacting domain cannot pull down tuberin, further suggesting that those two proteins can bind to each other. So what does tuberin do? There's quite a bit of recent literature out there that suggests that tuberin actually binds to hamartin, this other TSC gene product. And this complex is thought to, or the major function of this complex is thought to regulate a kinase, serine-threonine kinase by the name of mTOR. This goes through a small G protein called Rep. So TSC2 has a gap domain that can inactivate Rep. So what is mTOR? mTOR is a protein, a very critical regulator of cellular translation, and it can affect cell growth, survival, proliferation, and differentiation by phosphorylating a protein called S6 kinase and another one called 4-EBP, and these phosphorylation events essentially stimulate translation of certain messages. So our data had suggested that polycystin 1 might be upstream of tuberin, and we took a look to see whether mTOR itself might also be affected in those cells that express anti-MPC1, and we noticed that anti-MPC1 in controlled cells has again a sort of a diffuse cytoplasmic localization, but in cells that express anti-MPC1, now we see this very focal Golgi-like localization, suggesting that it might also be part of this complex. So if polycystin 1 would have a function in regulating mTOR activity, we would expect that in a patient where polycystin 1 is defective, that mTOR would be dysregulated. So to test this, we took an antibody against a phosphorylated version of mTOR, which is indicative of active mTOR. And we immunostained kidney tissues from ADPKD patients or normal human patients, or normal controls, and noticed a very high immunosignal here in the cyst lining epithelium in ADPKD, while there's very little immunosignal in normal tissue. So this suggests that, yes, indeed, there's a very high activity of mTOR in those cysts. We can also look at the downstream substrate that is phosphorylated by mTOR, in this case S6 kinase, using a phosphor-specific antibody, and get the same result. We see a very high level here in the cyst lining epithelium, very little in neighboring normal tubules or in a normal kidney. So because available human tissue usually represents end-stage disease, we try to see whether mouse models, where we see the same phenotype in mouse models, where we can catch this at an earlier stage. And we compare here, this picture shows 3 different mouse models. One is a conditionally inactivatable PKD1 model. So this is a very similar this very closely mimics the human disease, and we see the same result. In this case, looking at a phospho-S6, with phospho-S6 antibody, we see high levels in the cyst-lining epithelium, but also in 2 mouse models where very different genes are affected, namely MEL and a gene called ORPK. Even in these animals, we see high levels of mTOR activity in the cyst-lining epithelium, suggesting that the mTOR activation might be a common downstream pathway in all of these genotypes. So mTOR got its name by the drug that inhibits it. So it stands for the target of rapamycin. Rapamycin is a small organic molecule that had been discovered in the 1970s, and it was found that it actually inhibits very specifically and very effectively mTOR kinase activity. Rapamycin is also a clinically approved drug that is used as an immunosuppressant in kidney transplant patients, and we reason that if mTOR is dysregulated in polycystic kidney disease, we should be able to see a beneficial effect by treating with rapamycin. So that's what we tested in 2 mouse models that I'm showing you. So here I'm showing you one of those 2, the so-called ORPK rescue mouse model, which is a slow onset polycystic kidney disease model. We treat animals from postnatal day 150 to 178, so for 1 month. And the untreated mutant animals, as you can see, have a very highly cystic kidney. The non-mutant animals at the same age have no cystic kidneys here. And if we treat those animals for 1 month with rapamycin, we get much smaller cysts. And we can quantify this here, and it's a very significant effect. To test whether we essentially prevent the additional growth of the kidneys, or whether we can actually affect a regression of the kidney cysts, we did this experiment where we subjected mice to, so live animals, to MRI scanning using a special coil in a special facility. So we can measure the kidneys. So these are normal kidneys, those are polycystic kidneys. We can quantify the volume of those kidneys, and then measure the animals at day 150, 164, 178 during the course of the treatment. And as expected, a non-mutant animal does not show any change in the total kidney volume. Also, as expected, a mutant animal untreated shows an increase here. This is what we get with a mutant animal that is treated with rapamycin for 1 month. So not only does rapamycin prevent additional growth, it actually causes regression of the total renal volume. So one hypothesis how this regression could be achieved would be by induction of apoptosis. So we tested this. This here shows a tunnel staining here. So we essentially quantify the number of apoptotic cells. And what we noticed was that rapamycin treatment very significantly increases the number of apoptotic cells in the cyst lining epithelium. And we also see shed apoptotic cell fragments in the lumen there. Another mouse model that we looked at is a very aggressive early onset mouse model, the so-called BPK mouse. So here we have to treat the animals very early on from postnatal day 7 until 21 for 2 weeks. The mice typically die very shortly after postnatal day 21. And as you can see, day 21 old animals have very, very large kidneys, highly cystic in comparison to the non-mutant animals. If you treat them with rapamycin for 2 weeks, we get much smaller cysts and much smaller kidneys. And again, this is quantified here. This plots the kidney weight as a percentage of total body weight. The mutant animals have this very large spread here. If you treat them with rapamycin with 2 different doses, this goes down at more than halfway towards the control levels. And importantly, also we can measure the kidney function in those animals by measuring the blood urea nitrogen level, which accumulates in mutant animals. So the mutant animals show these very high levels, suggesting that they're short of renal failure. Treating them with rapamycin brings those values down to wild type levels, suggesting that we can actually prevent the loss of kidney function there. The final experiment that I'm going to show you here is a retrospective, very small clinical study that we did in collaboration with my clinical investigators at the Cleveland Clinic. So what we did is we took advantage of the fact that the many polycystic kidney patients will undergo kidney transplantation. And what the surgeons typically do is they add a third kidney into the patient. So without removing the polycystic kidneys, those patients then get a third kidney added to them. Those are the rare cases where a person has 3 kidneys. And those patients will be under immunosuppressive treatment from that moment on. And a group of those patients will be receiving rapamycin, which means those are people that have that are being treated with rapamycin, and they also have polycystic kidneys. So we quantified the renal volumes, the total volumes of those patients, essentially before and 2 years into the rapamycin treatment, to see whether rapamycin treatment made any difference. This here is the control group, not treated with rapamycin, but with other immunosuppressive agents. And these are the rapamycin treated patients, where we saw a significant decrease by about 25%, while there was no significant change in the control group. So this was a very small study. However, it points in the right direction, and it makes us cautiously optimistic that rapamycin might actually be effective in polycystic kidney patients. So finally, just our working model currently as to the mechanism of how polycystin 1 affects mTOR activity. So we think that our data is consistent with a model where polycystin 1 recruits a complex that induces essentially proximity between tuberin and mTOR. So as I told you, tuberin has the function of inhibiting mTOR via the small G protein Rep. And we think that this complex formation would essentially result in a constitutively inactive mTOR molecule. However, in an ADPKD patient where polycystin 1 is absent or defective, this complex could not form. mTOR would be able to be constitutively active and would do what we see it is doing there. A way to stop this essentially would be treating with rapamycin. So that's our working model right now. There are many aspects to it that we have not yet found out, but that we're working on. And just to acknowledge the people who have done the work, everybody who is in bold here with a star has contributed to this study. The 2 people that are currently in my lab here are Jonathan Schillingford and Senghui Lo, and those are the important collaborators from other institutions. Thank you very much.
We'll move to the final speaker of the first session, Mike Mahan, who will speak with us not on a genetic disease, but on infectious disease from the point of view of vaccine and vaccine development against microbial pathogens. Mike, like Dennis and Joel, received his undergraduate training at UC Davis. Were you guys roommates by any chance? What was in the water? Mike then did his PhD work at the University of Utah School of Medicine, where he undertook genetic studies and gene control in the microorganism Salmonella typhimurium. And from there moved to Harvard for his postdoctoral training in the area of microbial pathogenesis. And we then were fortunate enough to recruit Mike to join us on the faculty here in 1993 as an assistant professor. He rapidly rose through the ranks and now is a professor in the MCDB department, and also, as you've heard the thread through the afternoon, the BMSE program as well, another example of the cross-disciplinary research efforts that are ongoing. Mike will speak with us on vaccines, work that initiated, I believe, from studies in Salmonella and the DNA adenine methylase work, to then more broadly applied to not simply Salmonella, but also studies with other pathogens, the plague. And so the title of his presentation in vaccine research is Cross-Protective Vaccines Against Emerging Infectious Agents. And these will be microbial agents, not the avian flu, right?
Well, thanks for the opportunity to speak to you today about we think a very important topic. That's vaccine research. It's when one thinks about morbidity and mortality worldwide, second to water sanitation, vaccines are one of the best medical inventions of mankind. The focus of our lab is to understand the mechanisms by which bacterial pathogens cause disease, with the goal of utilizing this information to make medicines. And here's the leading causes of death worldwide. Infectious diseases, cardiovascular disease, and cancer contribute to the majority of deaths worldwide. And when I talk about cardiovascular disease, we're talking about heart attacks and strokes. Infectious diseases, many microbes can cause these, and also cancers. And what we're learning is that cardiovascular disease and cancers, many cardiovascular diseases and many cancers, have a microbial component. Some point in your lifetime, you've been exposed to a microbial agent that now manifests itself. It can manifest itself in a cancer or a cardiovascular disease. One of the principal examples of that is hepatitis B. Hepatitis B, if you're a person with hepatitis B and you get a chronic inflammation from the virus in the liver, it can lead to liver cancer. And now we immunize for hepatitis B, or immunize our children for hepatitis B. In fact, it can be considered the first vaccine against cancer, is the vaccine that we give to our children now. Here are the leading infectious diseases that cause the infectious disease deaths worldwide. Believe it or not, 80% of all infectious disease deaths are due to these 6 infectious diseases: respiratory infections, AIDS, diarrheal disease, TB, malaria, and measles. And respiratory infections, which is the leader every year, is basically the flu, pneumonia, things of that nature. The HIV virus is the causative agent of AIDS, and diarrheal diseases. In fact, diarrheal diseases are the leading killer of children worldwide. Every 15 seconds, a child dies of diarrheal disease, and that's going to be the subject of today's talk. I'm going to give you a little outline of what I hope to present today, is that we've identified a genetic switch called the DNA adenine methylase, or DAM, that controls bacterial virulence in several different agents. When we knock out the DNA adenine methylase, it completely disables the ability of the pathogen to cause disease. We've now utilized these DAM mutants to generate vaccines, cross-protective vaccines, that is, vaccines that protect against more than one strain. And such cross-protective vaccines have wide-ranging application clinically and also against biowarfare agents. And we'll get into that later. And last, I'm going to leave you some applications to other emerging diseases and biowarfare agents, and also some applications to vaccine development. For most of the talk, we're going to talk about Salmonella. And Salmonella causes a wide variety of diseases, ranging from food and blood poisoning to heart disease, typhoid fever. And so I'm going to give you a life cycle of Salmonella typhimurium, and that means mouse typhoid. So we have an animal model for typhoid fever. So how one gets typhoid fever: it's orally ingested. It survives the gastric barrier of the stomach, and then it now crosses the small intestine, where it's then taken up by macrophages. Salmonella have defense mechanisms that allow them to be resistant to killing by the macrophage, and then it allows them to basically use them as an immune tank to be able, or a taxi, to be able to now transport them to other positions in the body. First they go through the lymphatics, then they enter the bloodstream, where they disseminate into target organs such as the liver, the spleen, and the bone marrow. The numbers of Salmonella become quite large in these organs, leading to a septicemia. Salmonella can now, to complete the life cycle, can travel from the liver to the gallbladder through the bile, which is then dumped into the small intestine, leading to inflammation and ulceration. And the end point of this infection process is multiple organ failure and death. And what we're interested is understanding the virulence factors required for each stage of the infection process. And it's quite clear that Salmonella is not spewing out toxins at these various stages. In fact, virulence factors are some of the most tightly regulated genes. They're only made at the right place, at the right time, at the right quantity, because a pathogen always has to ask, is it worth it? Should I make this product? Because they're trying to avoid immune recognition. Okay. So one of the main obstacles to understanding microbial pathogenesis is that these bacteria behave like a Trojan horse. They hide a lot of their weapons until they're in an animal, and again, only at a specific time and place in the animal. And so we developed technology, or use this technology called IVET, which is in vivo expression technology, which simply put allows us to select for bacterial genes that are on in a mouse and off out of the mouse. And we've identified many of the known Salmonella virulence functions through this technology. But for today's purpose, I'm going to tell you about how we've now utilized this technology, this ability to select for genes that are on in a mouse and off out of the mouse, and look for mutants that disable this switch. Because if we can disable this switch, we may theoretically be able to disable the ability of a pathogen to cause disease. So we did a simple genetic test experiment where we now look for mutants that now, again, disable the switch. So we start with bacterial genes that are on in a mouse and off on the plate. And then we now look for mutants that basically are now on in the mouse and on the plate. These are tightly regulated, these are unregulated. And this allowed us to uncover the DNA-adding methylase. Lack of the DNA-adding methylase allows expression of many of these virulence functions and immunologic functions. So we made this discovery. It was appealing to us because David Lowe, who's right down the hall from me, had shown that the DNA-adding methylase was very important in urinary tract infections. It's involved in the formation of these pili, these extensions that emanate from the bacteria and allow the uropathogenic E. coli to do a very special thing. It allows them to be able to ratchet up like a mountain climber up the urinary tract. It's not good enough just to bind. You got to get up into the bladder, into the kidneys. You have to attach and detach, just like a mountain climber. And the DNA-adding methylase he had shown was involved in the switching on and off of these pili. So then we tested regarding if the DNA-adding methylase really indeed have an effect on virulence. And so now we're talking about Salmonella virulence, we're talking about typhoid fever. Okay? It takes about 100,000 organisms for the LD50 for typhoid in mice. When we now knock out the DNA-adding methylase, the LD50 rises over 10,000-fold. In fact, the mice, not only the mice don't die, they show no symptoms whatsoever of being infected with this dose. We then overproduced the DNA-adding methylase, and we got the same result. That basically leads to very, very marked defects in virulence. So the conclusion from this experiment, either the lack or overproduction of the DNA-adding methylase leads to a marked reduction in virulence. Okay, now I told you at the beginning that when we want to make medicines, okay, and the medicines that we make are vaccines. And so the idea was that here we have these live organisms that are not causing disease. Can we now use these as vaccine candidates to elicit immunity? Okay, and I'm going to tell you that we now immunize with the DAM- mutants. Well, first we now challenge non-vaccinated mice. Here are the orange or yellow squares here, with 10,000 times the LD50, and it kills all mice, all chicks, all calves that we now tested. We now immunize with the DNA-adding methylase mutant, and now challenge with the virulent strain, we get 100% protection in mice and chicks at 10,000 times the LD50, and a significant protection in calves. So the conclusion for this experiment is yes, DAM- vaccines are live vaccine candidates. Okay, now how can the loss of one enzyme completely disable the ability of a pathogen to cause disease and elicit these robust immune responses? And so what we decided to do is just look at the colonization ability of the vaccine strain compared to the virulent strain. And so we'll go through this a little bit slow here. So basically the open squares is the wild type virulent strain that causes typhoid. We now orally infect mice and simply look and count where the bacteria are going. Okay? So on day 1, so I'll go through this first. These are the Peyer's patches. PP stands for Peyer's patches. They're lymphoid organs that line the intestine, basically that have all the immune cells required to elicit a robust immune response. Basically, they're our portal to the lumen of the immune system. They simply look, they see what antigens we're being exposed to, and decide, should we now generate an immune response. They line the Peyer's patches. MLNs and mesenteric lymph nodes, and then the liver and the spleen. So these are your immune organs. So now we orally infect with the wild type, and now we just follow it. So in day 1, we find at about 10 to the 4th to 10 to the 5th wild type bacteria in the Peyer's patches, mesenteric lymph nodes, liver, and spleen. We now wait 5 days, and you can see that the wild type organisms remain the same in the Peyer's patches, but then now they've crossed over into mesenteric lymph nodes, and they're proliferating in the liver and the spleen, and there's no day 6 for these mice. They die of typhoid. Okay? Well, something very different happens when we use the live vaccine, the live dam-minus vaccine. At day 1, we get colonization at numbers similar to wild type. We show a defect and be able to colonize the mesenteric lymph nodes in the liver and the spleen. We wait 5 days, and the live vaccine is still colonizing the Peyer's patches. These are the cells you need to elicit an immune response. We show a defect in the ability to reside in the mesenteric lymph node, and we don't find this live vaccine in the liver and the spleen. So our conclusion from the experiment is that because the Salmonella target the Peyer's patches, in fact, that's what the wild-type Salmonella does. It takes out your Peyer's patches, okay? So we've used this innate ability of the pathogen to be able to target the appropriate immune cells to elicit a robust immune response. However, it's defective in survival and replication in systemic tissues, and that may explain the reason why these vaccines don't elicit disease manifestations in these mice. So to get into the molecular basis of what's going on here, and this will be short. Basically, we wanted to see what we were thinking was, well, have we hit a genetic switch that now results in the elaboration of antigens, elaboration of immunogens that now elicit these robust immune responses? And the answer appears to be yes. Here's it's a very small portion of what's called a two-dimensional gel. It's a way to look at all the proteins that are produced in a bacterial cell. And here we have the wild type, the dam-minus mutant, and the dam overproducer. What we have with these arrows here are the production of a protein. We don't know what the protein is at enhanced levels in the dam-minus mutant. And in fact, we've identified several proteins that the dam-minus mutant makes what we call ectopically at a very large level. We've also shown, it's not shown in this slide, but with the dam overproducing mutant, a different set of antigens or different set of proteins are now are ectopically produced in these cells. And what we hypothesize is that now we're looking at a very, very large repertoire of antigens that are not even elicited in a natural infection. And I can tell you, I didn't bring the data, but the level of immunity that is elicited by these vaccines is much more than survival from a natural infection. Okay, so now this is the bones of the talk about the issue of cross-protection. Vaccines are great. They work great, but they have an Achilles heel, and the Achilles heel is they only work in the strain that they're made in. Okay, there's no what's called cross-protection, one vaccine protecting against several strains. This is why you have flu vaccines every year. There's a different strain of the flu. This is why you can get epidemics in previously immune populations, because a different strain is now infecting. Okay, and so this is a real problem in Salmonella. There's 2,500 different pathogenic strains of Salmonella. That's why there's no vaccine. Okay, this is a huge, we're talking about California, it's a huge cost to our agriculture in the state. We have a very large chicken producer, egg producer, largest dairy producing state in the nation. We make a lot of milk and a lot of cheese here. Okay, nothing on Wisconsin, but you're number 2. Okay, so now we're going to talk about cross-protective vaccines. Okay, now we're going to just talk about 3 serotypes. We're now doing this in a much larger bit with this slide. We want to talk about 3 serotypes of Salmonella, 3 strains of Salmonella. This is Salmonella typhimurium, the strain that causes mouse typhoid. This is Salmonella enteritidis. These 2 strains are the 2 leading causes of Salmonella infection in humans. Okay, and basically how you get Salmonella, it's pretty uncomplicated. You're eating contaminated chicken or contaminated eggs. Okay? And we've also decided to test Salmonella Dublin, which is a problem in calves, and it's a big problem in the state of California. Okay? So now what we're going to do, we're now going to immunize with one strain of Salmonella and now see if it protects against the other strains of Salmonella. Okay, so if you're non-vaccinated 1,000 times the LD50, all of the animals die. And if you now immunize with a Salmonella typhimurium, you get 100% protection against the homologous strain, the strain that the vaccine was generated in, and you also get significant protection in Salmonella enteritis and Salmonella Dublin. We've now also overproduced the DNA editing methylase and get a similar result. Immunization with Salmonella typhimurium results in a cross-protective response to Salmonella Dublin. And so the conclusion with this experiment is, yes, you can generate vaccines that elicit cross-protective responses to more than one strain. And this is paramount to our defense against biowarfare agents. I mean, there could be multiple strains released. That's going to override the immunity of any single vaccine that we have today. We don't have cross-protective vaccines. 2, they can now actually select for super strains in vaccinated animals, okay, that will make our current vaccines obsolete. So this is something that we need to solve, and we need to solve it fast. Okay, so is this technology only available or only applicable to Salmonella? We have the DNA editing methylases in many bacterial pathogens. There's a short list just for you to look at. The plague, typhoid, dysentery, cholera, you can go down the line. This is just a graphic representation. So our next thing that we wanted to do was say, okay, can we now use this technology in another pathogen, another important pathogen, and we've chosen Yersinia, the causative agent of the plague. And I can tell you that Yersinia is a very different pathogen than Salmonella. Salmonella likes to be inside cells. Yersinia likes to be outside cells. And this is why we kind of chose, and also with the issue of it's a top 5 biowarfare agent. And it's how we're funded. Okay. So this is very similar to Salmonella. We overproduced the DNA editing methylase in Yersinia, the plague-causing organism. This is in mice. And again, when we now overproduce the DNA editing methylase, we get a marked reduction in virulence over 6,000-fold, just like Salmonella. We now take these immunized animals, and we now challenge them with the virulent strain, just like I told you before. And we get 100% protection at 1,000 times the LD50 against mouse plague. And so what we're now doing is that we're now trying to test whether this vaccine can elicit cross-protective responses to several types of Yersinia, and that'll be the focus of our work in the future regarding biowarfare. Here's our working model of how the role of DAM in virulence. I mean, and how does, again, the alteration of one enzyme lead to ectopic gene expression, defects in virulence, and elicitation of these robust immune responses. And if we think the answer might be simple, or at least the general answer might be simple, we believe it's because its global role in bacterial gene expression. Okay, and an example for the wild type here, the wild type, the virulence and immunologic functions are tightly regulated. Okay, and these functions and the actual functions now change as infection proceeds through the infection. Okay. This is not what happens in the DAM minus or DAM overproducing mutant. We are topically producing several functions. Okay, so due to aberrant gene expression, this leads to defects in virulence and the elicitation of these robust immune responses. So where are we going with this technology? We're now using this DAM technology in several different pathogens, and again, we're trying to focus on the cross-protection that can be elicited by these pathogens, and I think we've demonstrated the feasibility that yes, you can develop cross-protective vaccines. We're also using this technology, this DAM technology, as a platform to be able to express heterologous antigens from other microbes, other viruses, other bacteria, to be able to elicit immune responses to them. And I'll be quite frank, we don't know exactly what we've done, what are the immune responses. We're now working on that. Okay, but we have high hopes that we can use this technology to be able to elicit responses from other pathogens. And last, this leads to research in small molecule inhibitors that now affect this enzyme, and also other players in the pathway. I'd like to conclude this talk with a couple people I'd like to acknowledge. A lot of them are at UCSB. Doug Heitoff runs my lab. Steve Giulio, who now is going to be a professor actually at Westmont. And Bob Sinsheimer, and David Lowe. These people had the most, for the talk I'm talking about today, they had the most input, and also with a lot of the chick and cattle work is being done in Sydney, Australia. And I thank you for your attention. That was a lot of fun.